If you come up to my office, I may have these things. I should have. I hope I have them all graded by month. Okay, but I can't really. Yeah, that's the intention. These things take a while. All right, we're ready for photosynthesis. Come by and I can tell you. I can't pass it back. I can tell you how you did if I've got them graded. In fact, I've got, like I said, I've got about half of them graded now. So if you want to come by my office later on, I can tell you what you got and stuff like that. All right. Okay, well, photosynthesis. Clearly one of the most important breakthroughs in the history of life on this planet. We'll look at that a little bit more towards the end of it. But photosynthesis allows you to use the energy of light, sunlight, and simple stuff like carbon dioxide and water, and ultimately produce your own organic molecules, specifically glucose. And you can save that stuff, and when the lights go out, metabolize it later by glycolysis, by fermentation, by oxidative metabolism, all that kind of stuff. So they call photosynthetic organisms autotrophs, literally self-feeding. They make their own food from simple stuff that's common in the environment. Okay, photosynthesis first got its start in bacteria. We have plenty of photosynthetic bacteria. They call them cyano, blue-green bacteria. It's blue-green pond scum. Those are photosynthetic bacteria. There are other bacteria that photosynthesize, but they do it differently. They use things like hydrogen sulfide instead of water, or metal ions and things like that. Those are oddballs that live in really extreme environments, and they were probably the first photosynthesizers on this planet. But more on that later. But the vast majority of photosynthetic organisms, bacteria and plants and photosynthetic protists, do it the way that we're going to be showing fairly soon. Now, photosynthesis in bacteria, you have some processes occurring on their cell membranes, specifically the pigment complexes that capture light, and you have some stuff occurring in the cytoplasm. But this is, of course, on eukaryotic cell biology. So in eukaryotes, this process occurs in specialized organelles, the chloroplasts. And we'll take a look at these guys later. They're very similar to mitochondria, and once again, they're very similar to photosynthetic bacteria. That's where they eventually evolved from. Endosymbiosis, photosynthetic bacteria inhabiting ancestor eukaryotic cells to the benefit of both. We'll see that again. It's very similar to what we see in mitochondria. Okay, so it occurs on specialized organelles in eukaryotic cells. But photosynthesis consists of two major things. What we often call the light reactions, or light-dependent reactions. I think that's what the book uses. And they're called that because they have to use light. That's the source of energy. This is where you have the chlorophyll and other pigments. Light gets absorbed. Electrons get kicked off the pigments, passed down electron transport chain, just like what we see in mitochondria, folks. And the energy of these oxidation reduction reactions is ultimately used to produce two energy-rich molecules, ATP and one of these nucleotide coenzymes, NADPH. It's called NADPH because it has an extra phosphorus compared to NADH. That's why they throw the P in. So these are energy-rich molecules. And light is absolutely required for this to happen. That's the source of energy to produce these molecules. So in a sense, it's like a power factory, the power plant of the chloroplast. Now, the next thing is the chemical synthesis, what we sometimes call the light-independent or dark reactions, because they can occur in the dark as long as you have the right ingredients. And most of that consists of what's known as the Kelvin cycle, named after the especially if I can spell the word cycle here. Okay, named after the biochemist Melvin Kelvin, who mapped it out decades ago, got a Nobel Prize for that, was active in research all the way to his late 90s when he just croaked one day. Okay, anyway, he got a Nobel for this work. But anyway, the dark reactions. This is where we're going to do the synthesis. We're going to take 6 carbon dioxide, 18 ATPs, it takes a lot of energy to do this, all 12 of our NADPHs, especially when it's Vellerite, and we make a glucose out of this. This glucose we can use later on when the lights go out, the same kind of metabolic strategies we use. Okay, so in a sense, you got these two parts. The power plant, that's the light reactions, and the chemical factory, that's the dark reactions. Now, if anybody's ever gone up north, if you ever drive up I-95, past into Delaware and southern New Jersey, the place looks like a seen-out Blade Runner. You have these huge factories, chemical factories. You know, Delaware is practically owned by the DuPont family, the chemical guys. And you see these enormous factories spewing out all kinds of nasty smells and multicolored smoke and fun things like that. And then you see these converging onto it, all these high tension wires, because you need a lot of electricity to power these chemical plants. In a sense, that's what we got here. We've got a power plant, the light reactions. We got a chemical factory that's a lot more innocuous than stuff DuPont makes, a lot less polluting, that makes our glucose. So that's what we see in the case of photosynthesis. Now, the next thing is let's take a look at these organelles, the chloroplasts, which is where this occurs. By the way, anybody have any questions? This is just a real skeletal outline here. Okay, let's look at the chloroplasts, the organelles. Map out what happens here. Chloroplasts are very similar to mitochondria in many respects. And like mitochondria, this evidence is very strong that this is an endosymbiotic relation, or at least the descendant of that. In other words, that chloroplasts were originally cyanobacteria, blue-green bacteria that got into certain ancestral eukaryotic cells some 2.5 billion years ago, and through horizontal gene transfer and stuff like that, they lost their cell wall, they lost their independence, and now they're part of the host cells. And of course, it works to the benefit of both, because now these cells can photosynthesize, they can get the sugar produced by photosynthesis, thanks to their little bacterial companions. And the other cool thing is that cyanobacteria, or former cyanobacteria, are much more protected because they're inside a much larger cell that can move around and go to the sunlight. So it works to the benefit of both. Okay, now let's see what chloroplasts look like. Like mitochondria, chloroplasts have a double membrane with the inner membrane designed to increase surface area as much as possible. But it's going to be different than the mitochondria. We just fold up the inner membrane to those folds called cristae. Here we're going to do something a little bit different. So let's take a look. Road map the chloroplasts. We have, first of all, an outer membrane. Once again, is specialized for transport, getting things in and out. Okay, now we have an inner membrane separated by brief space. We don't have to worry about that space between the membranes like we did in the mitochondria. It's just there, but it doesn't have any real functional significance. You recall for the mitochondria, we pump H plus over into here. We're not going to do that in chloroplasts. We will pump H plus, but not into the same place. Now, to increase surface area here, what we're going to do, instead of folding up the inner membrane, is we branch it off, and then we develop these series of stacked sacs, little oval sacs of membrane. And we call these stacks the grana. The individual sacs are called the thylakoids. Okay, and we continue this on. We do this over and over again. I'm just going to put a few here. In real life, of course, the chloroplast is really packed full of these thylakoids. I'm just doing a little bit here so we have some interior space. But like I say, it's much more densely packed than what we see here. Once again, the idea is to put as much surface area of that thylakoid membrane as you can fit inside the small chloroplast. So here we got thylakoids all over the place. And we continue it on and so on. Okay. All right, here we go. All right, so it's really packed full of thylakoids. That tells you that there must be something really important about those thylakoids, because you want to pack as much of that membrane into the chloroplast as possible. So what goes on in the thylakoids? What's going on in the thylakoid membrane? The thylakoid membrane, folks, is the home of the light reactions. All the goodies, all the components we need for the light reactions are embedded in the thylakoid membrane. And that includes a number of things which I'm going to map out in a second. That includes what are known as the photosystems. The photosystems are complexes of proteins and enzymes and numerous pigment molecules. Chlorophylls, luteins, xanthins, all kinds of different stuff. Chlorophylls, of course, being the major one, the green stuff. So the photosystems are the pigment and protein complexes that actually absorb the light. We have associated with these photosystems electron transport chains. Okay, because when we absorb light, we're going to knock electrons off these chlorophylls, pass them down electron transport chains, a series of oxidation reduction reactions. That energy gets released. Now we can do something useful with it, like make ATP or make NADPH. And then, okay, and then we have ATP synthetases. The ATP synthetase of chloroplasts is another magic mushroom. It's very similar, not quite identical, but it's very similar to the mitochondrial ATPase. There's a few slight differences, but works the same. We'll see that a little bit later on. So that's the thylakoid membranes. This is the home of the light reactions. Now, to make ATP, we're going to, once again, pump H+ ions across a membrane into an area of high concentration. Active transport of H+ ions. In mitochondria, we pumped it into the space between the inner and outer membranes. Here, we're going to pump it into the interior of the thylakoids. So let's put that down here. We'll put thylakoid interior. That's where we're going to pump the H+. We're going to pump it across the thylakoid membrane into the thylakoid sac itself. So these thylakoid sacs are bags full of acid, about pH 5.5. That's about 80-fold concentration difference. And that's going to give us the power to run that, let it leak back through ATP synthetase, and therefore make ATP. It's the same chemiosmotic mechanism that we saw in mitochondria. You actively transport H+ into an area of high concentration. You facilitate diffusion of the H+ through the ATP synthetase. That energy release powers the actual production of ATP. We described the little whirligig, the little miniature electric motor mechanism that these guys have. Okay, so that's the thylakoids. Clearly, the thylakoids are very important. That's why we pack as much surface area thylakoid membrane in as possible. The more of these photosystems, the more ATP synthetases and stuff you can pack into chloroplast, the more efficient photosynthesis is. And when you come down to it, it's a good thing for plants that they don't move around a lot. Plant cells use a lot less energy for the most part than animal cells. If you actually had a little green man, a photosynthetic organism like a person or something, photosynthesis would not be able to provide enough energy for what we would consider normal human activity. You just wouldn't have enough surface area to pick up light. But if you're a plant, you don't run around and things like that. You operate in slow motion. So you can get away with photosynthesis on its own if you happen to be a plant or a cyanobacteria. But it wouldn't work too well for animals. It could supplement their energy, but it wouldn't be able to provide enough for highly active animals like ourselves. Okay, well, we got that. Now, the fluid-filled interior. This is the equivalent of the matrix of the mitochondria. But biologists don't like to use the same words over again, so they're going to call it the stroma instead of the matrix. It's the same thing, basically. It's a fluid-filled interior. Now, what's in the stroma? Important stuff. We're going to have the dark reactions, the Kelvin cycle and something in some plants called the C4 cycle. More on that later. Inside the stroma. Once again, these are multi-step enzyme cycles, kind of like Krebs cycle, but a different kind of function. And this is where we actually produce the glucose from carbon dioxide, ATP, and NADPH. So we're going to have the dark reactions are here. Another thing we got in the stroma, just like what we see in mitochondria, chloroplasts have their own DNA in the form of one or more, usually multiple copies of a circular bacterial-like DNA molecule. And chloroplast DNA, the circles are larger than what we find in mitochondria. They tend to code for about 50 to 100 genes instead of about 20 to 40 in mitochondria. So they actually make more of their proteins, about 5% of their proteins compared to the couple percent that mitochondria make. So they're a little bit further along. They still make more of their proteins. So circular DNA. Okay. Plus, of course, they have their smallish prokaryote type ribosomes in there to synthesize these proteins. The ribosomes, once again, are very much like the prokaryotic ones. They're distinctly different from the ribosomes in the cytoplasm of these cells. They're sensitive to the same kinds of antibiotics like streptomycin stuff that bacterial ribosomes are like. Once again, biochemically, genetically, all this stuff points to bacterial ancestry. Specifically, they sequence the genomes of these chloroplast DNAs, and it puts them into a class of cyanobacteria. The closest relatives are a certain group of cyanobacteria. So that was their ancestors. Okay, so that maps out the chloroplast. That's where these things are happening. Okay, questions on that? Roadmap. Once again, test questions will often include processes and locations of those processes. Okay, you.
At the top, is that, what's the G word?
Oh, grana, G-R-A-N-A, and that says in parentheses, stacks of these thylakoids. Yeah, question back there.
Bottom bullet.
Bottom bullet, small prokaryotic type ribosomes. In other words, like bacterial ribosomes. Okay, great. All right, so that's a roadmap, folks. Now let's take a look at what's going on here. We're going to start out by looking at the light reactions. And before we do that, we are going to first introduce our major character. Because good stop point. We'll do another video. All right, let me just stop the video and read. Why are you messing me down? Okay.